Hello, everyone. I'm Kyung-Yeop Kong, breast pathologist from Asan Medical Center, Seoul, Korea. Today, I'll interview with Professor Kirsten Dankert regarding immune microenvironment of breast cancer in immunotherapy session GBCC 10. Hello, Dr. Dankert.
Hello, it's a pleasure for me to participate.
It is great pleasure to talk to you in precision session. Though you are well known to most breast cancer specialists with your works, including myself, as I've started my current research on TILs 10 years ago, inspired by your JCO paper. Could you introduce yourself briefly to the viewers?
Yes, so I'm a professor of pathology at the University of Marburg, which is located in the center of Germany. I have been studying breast cancer for many, many years, and we are working in research together with the GBG, the German Breast Group, which is a major clinical trial group in Germany.
Okay, let's move on to the main topics. You are planned to give a lecture about immune microenvironment of breast cancer in GBCC at April this year. You would like to discuss about several issues related to your upcoming lecture. I'll ask the first question. Is there some distinct characteristics according to molecular subtypes of breast cancer in terms of immune microenvironment?
Yes, I think it's a very important message that we learned from daily clinical pathology, that breast cancer are actually many different subtypes, and those subtypes can be molecularly defined. And what we observed is that there are particular subtypes like the triple negative tumors and the HER2 positive tumors, where we have subsets of tumors that have a large degree of immune activation, which we, for example, can observe as increased TIL infiltrates.
Okay. It seems that there are agreements among researchers about TNBC and HER2-positive breast cancer. But about the luminal subtypes, what is your opinion about their characteristics of microenvironment?
Yes, I think there are still really a lot of research options in the luminal breast cancer field. So as you said, for TNBC and HER2 positive breast cancer, I think it's quite clear. For luminals, well, we have many tumors that have a comparably good prognosis, that are slowly proliferating, and we do not see much immune activation in those tumors. But on the other hand, there are still luminal tumors in the luminal B type, where the tumor is more aggressive. We see a lot of Ki67 staining. And I think those tumors would be very interesting to investigate for immuno-oncology questions. And it would probably be also interesting to include them in ongoing or future clinical trials in this field, because I think there are a lot of good therapy options for those tumors.
Okay, I see. So next, do you think there are differences in immune microenvironment of breast cancer compared to other solid tumors? And if so, what differences exist and what makes the difference?
Yeah, this is actually a very, very complex question, because it's difficult to compare different tumor entities. I think what we know for immuno-oncology is that there are some very typical examples of tumors like melanoma or some types of lung cancer, where we have a clear relationship or clear contribution of the immune environment to the control of the tumor. And those are typically tumors that have a distinct mode of tumorigenesis, like sunlight for melanoma or smoking for lung cancer. For breast cancer, I think we have the situation that there are many, many different options how those tumors can be generated. So we do not have we have some tumors which are due to dysregulation of hormone pathways. We have some tumors that have a higher degree of genomic instabilities. Some are caused in the BRCA background, and I think this makes it very complex. So I think the immune environment is important for some tumor subsets, but it's much more diverse in the field of breast cancer compared to other types of tumors.
Okay. And next, recently, neoadjuvant treatment for breast cancer patients is widely accepted. Is there some notable change in immune microenvironment before and after the neoadjuvant treatment? You published a lot of articles recently.
Yes, so I think we all learned that the neoadjuvant situation is an extremely interesting field also for immuno-oncology, because many groups have observed that if there is a high immune activation in a tumor, neoadjuvant therapy will work better. So what does it mean for the residual tumor? First, many tumors that have a high immune infiltrate have a complete response, so we will not see them in the residual tumor anyway, and those patients have a good prognosis. But there are still some tumors remaining, and we are right now doing a large project to characterize different types of residual tumor. And what we see is that there are tumors that still have an immune infiltrate, and there are reports that those tumors still have a comparably better prognosis compared to those tumors that are completely resistant to immune activation, that do not show any immune infiltrate. But I think this is really a very active field of research, and we need to learn a lot about the different patterns of tumor regression in the neoadjuvant situation.
So do you think the status of my immune microenvironment after neoadjuvant treatment is related to prognosis of patients?
Yes, where we know that generally if there is tumor left after neoadjuvant therapy, this is associated with a worse prognosis. So there are considerations to do post-neoadjuvant therapies in this situation. But nevertheless, those subgroup where there are still immune cells present in the residual tumor, they have a slightly better prognosis compared to the other ones.
Okay, thank you. Could you briefly explain about the currently available modalities to evaluate the immune microenvironment, and also considering real-world daily practice in the medical center, what is the most feasible method to recommend?
Yeah, so I think a very first step that can be done in any pathology assessment of any core biopsy or breast cancer specimen is simply the evaluation of TILs, of tumor infiltrating lymphocytes. So there are many immune cells present in the tumor microenvironment, and then they can be very easily observed on an H&E section. And together with the TIL working group, we have defined the best way to evaluate those TILs. So we are focusing on the stromal TILs, which are in the tumor stroma next to the tumor cells, because they are the best parameter. And there are many ring trials that this can be used as a very good first approach. Then, as a next step for research, obviously, it's important to characterize the immune infiltrate on a molecular level. And a very good approach to do this, in addition to immunohistochemistry staining, would be mRNA signatures, where we can detect typical mRNA signals for the different types of immune cells. And I think the combination of all those methods will be the best approach for future research.
Okay, as a pathologist, I totally, absolutely agree with your opinion. TIL is a very good choice. Okay, so nowadays in Korea, digital pathology is a hot issue among Korean pathologists. Do you have a personal experience in applying digital pathology to evaluate immune microenvironment, including immunohistochemistry?
Yes, I think that pathology is moving from microscopes more to the field of computer science actually today. So there are many activities. We have performed some studies to identify immune cells on H&E sections, which is generally quite feasible because they have a very distinct morphology that can be detected by a computer algorithm. There are some options to do multi-level immunohistochemistry to determine the different cells in the immune microenvironment. And the advantage is that in this approach, we will still have the spatial resolution, so we can investigate the spatial distances of different types of tumor cells and immune cells. The problem, I think, for daily pathology is that this will generate very, very large data sets with information on basically all single cells. And this is difficult to do in a diagnostic situation, but obviously it's very interesting in a research situation, and we are doing some projects in this field.
Next, have you tried spatial genomics? Is new techniques, including nanostring digital spatial profiler or Helios system?
Yes, we actually do have a nanostring GeoMX project, which is ongoing, where we have generated a tissue microarray from immunotherapy clinical trials with biopsies before and after the therapy. And the advantage is that we get a spatial resolution of the mRNA profile, which can combine the spatial world of pathology with the genomic world. And we are expecting the results from this project within the next 2 months, and we think it will be very interesting.
Okay, currently immune checkpoint inhibitors are being introduced into standard practice of breast cancer treatment, and evaluation of tumor immune microenvironment will be more important as they can serve as predictive biomarkers. Do you have any opinion to give for better evaluation of immune microenvironment for immunotherapy decision?
Yes, I think the most important message actually was generated some years ago, where many pathologists observed that breast cancer is actually an immunogenic subtype, or that there are some tumors that are immunogenic, and that was a kind of additional starting point to evaluate those therapies in breast cancer. In the biomarker field, I think there are two important observations. The first observation is, if we look at the neoadjuvant situation, any type of immune activation will increase the response rate to therapy. And this is relevant for classical chemotherapy, but it's also relevant for immunotherapy. So if you give immunotherapy in addition to chemotherapy, the response rates will be increased. But it's not dependent, the effect of the immunotherapy is not dependent on the level of immune markers like PD-L1 or TILs. So in any situation, any type of therapy will work better if there are many immune cells or evidence of immune activation present. So this is in the neoadjuvant situation for early breast cancer. In the metastatic situation, it's actually different. In the metastatic situation, we have clear results from, for example, the IMpassion trials, that if we see levels of PD-L1 in the tumor tissue, those are the patients that respond, that benefit from the additional immunotherapy. And I think the best explanation for this difference between the early and the late situation is that we have selected for more aggressive tumors if we do a study in the metastatic setting. And if we still see some immune activation in those tumors, this is a positive predictive factor for therapy. And in the early situations, there are many tumors that can be comparably easily treated by different types of therapy. So we do not have this selection for a biomarker population.
Okay, so routinely you do PD-L1 evaluation in immune checkpoint inhibitor treatment?
So we do it in the GBG clinical trials, in the neoadjuvant trials, to generate data sets, additional data sets in the early situation. And we do it as a part of routine diagnostics in metastatic TNBC to define the best type of therapy.
Okay, so there are several kinds of PD-L1 immunostaining. So what kind of PD-L1 staining do you use?
So in Europe, generally, we are basically free to use any antibody that is validated in the laboratory. So we do not have a situation where it is required from the authorities to use a specific antibody for a specific substance. For triple negative breast cancer, for an atezolizumab therapy, the SP142 antibody is the antibody that is recommended. We use it in the clinical trial setting, and we also use it in the diagnostic settings, but other antibodies are also used in parallel.
Okay, I see. So we do similar staining. So for metastatic tezolizumab, SP142, we stain and report. So another treatment issue, from the first attempt of using TILs for adaptive cellular therapy for melanoma patients since the Rosenberg group in the 1990s, TIL adaptive cell therapy is being widely studied in different kinds of solid tumors. What do you think of developing TIL adaptive cell therapy in breast cancer?
So I think it's really an exciting question, and to move from TILs as a diagnostic marker to TILs as an additional therapy options. I'm not an immunotherapy expert, so I cannot explain or evaluate details of adaptive TIL therapy. From my message from pathology would be that it's probably very important to select the right tumor type, because if you just test any breast cancer, there may be some responses, there may also be no responses, but if you would focus on the correct subtypes of breast cancer where we see this is a tumor that has evidence of some immune activation, that maybe has a high mutational load or many TILs in environment or a high PD-L1 expression, I think those subtypes would be the most interesting for those new experimental therapies.
Okay, so any other additional comments for the viewers?
A pleasure for me to be invited as a speaker and to be able to participate in this conference. So I'm very happy about this.
Okay, many thanks for joining us today. It will be helpful for the people in studying immune microenvironment of breast cancer. I really hope to see you in Seoul, April. Okay, bye.
Yes, thank you very much.